Okay, hello everyone. My name's Tim Luca from Eli Lilly. I'd like to welcome you to the Sachs Neuroscience Investor Roundtable. We've got an esteemed cast of global investors and pharma companies involved in this panel, and it'll be chaired by myself and Jay Olson. So hopefully we'll get to the bottom of what's exciting in investing in the CNS space. So I think we're going to start off with a few brief introductions. My name's Tim Luca. So I do search and evaluation and venture investing for Eli Lilly. We've invested and acquired and collaborated with a lot of neuroscience companies, many of which are the sorts of companies that are created by many of the people on this panel today, for which we're very grateful, and I'm sure patients are very grateful too. So it's a very exciting space, and I've been in the industry for about 25 years. I've worked in big pharma, medium pharma, spun out biotech companies, and I'm a scientist by training, PhD scientist. So that's enough about me. I'll hand over to Jay.
Thank you, Tim. Welcome, everyone. I'm Jay Olson. I'm a sell-side biotech stock analyst at Oppenheimer in New York City. I cover a number of companies doing work in neuroscience, and it's a great pleasure to host this panel today. And I want to thank all of our distinguished panelists for sharing their expertise with us today. Really excited about the discussion. A lot of interesting things going on in neuroscience. My background is I originally worked in industry. I spent 16 years at Pfizer back in the days when Pfizer was involved in neuroscience, and mostly did marketing and business development. And then I spent the last 14 years or so doing sell-side equity research at Sanford Bernstein and Goldman Sachs and now Oppenheimer. And again, it's a real pleasure to be here today, and I want to thank everyone for joining us. And with that, we'll get some introductions from our panelists. Guy, would you like to go next?
Thanks, Jay. Good afternoon, everyone. My name is Guy Seabrook. I'm the neuroscience lead for the external innovation team based in South San Francisco. I'm an electrophysiologist by training. I've worked in industry most of my career. And at Johnson & Johnson, we're interested in areas of research associated with neurodegenerative diseases as well as neuropsychiatry. And so we work very closely with our Johnson & Johnson Development Corporation, which is the equity investing arm of our business, as well as the JLabs team that has a no strings attached incubator at various sites dotted around the globe, which house new biotechs, some of which we've invested in and others we've not, but where we forge long-term relationships with these companies and entities. So it's a pleasure to be here today, and look forward to sharing some of the perspectives on what's new and what's the future for CNS research and investing.
Great, thank you, Guy. Jamil?
Thanks, Jay. Good morning, good afternoon, good evening, everybody, depending on where in the world you're watching the conference from. Thanks for having me on the panel. Thanks for the invitation, and always thanks to Sachs for organizing. So Jamil Beg, partner at 5AM Ventures. I've been with the fund about 4 years now. Overall, about 15 years in the biopharma industry, about half on the operating side, mostly in small biotech startups, and the other half in investing roles. Just before 5AM, I was at Sage Therapeutics, a small molecule neuroscience company here in the Cambridge, Massachusetts area. Joined early on, less than 10 employees, helped to build the company up to over 200 employees and move the programs from discovery stage into several registration studies. 5AM Ventures, as the name implies, is an early stage life science venture fund. We've been investing now almost 2 decades, about $2 billion under management, 90 plus companies we've invested in over the years, and about 15 or so approved products over that time. Currently investing our 6th fund. It's a $350 million fund, and we'll be announcing our 7th fund later this year. We're therapeutic area agnostic, modality agnostic, invest quite heavily on the advanced modality side, cell and gene therapy. We're seed and Series A investors. We're always leading or co-leading investments, always syndicating our Series A financings, and sometimes syndicating our seed financings as well. And we really see ourselves as partners with entrepreneurs and academics to help build companies. We've got a long track record in investing in neuroscience, and I think every investor on the panel we've co-invested with. So great to see a lot of familiar faces.
Thanks, Jamil. Peter?
Well, glad to be here, and thanks to Sachs for organizing this. I'm Peter Thompson. I'm an immunologist and medical oncologist by training. I serve as an affiliate professor of neurosurgery at the University of Washington, and I'm a partner at Orbimed Advisors. I've been in the biotech industry, I guess, since the late Bronze Age. Perhaps founded about 6 or 7 or 8 companies. I'm a little unclear on how many at this point. Orbimed Advisors is probably known to some of the folks in the audience. We're the world's largest dedicated healthcare investor. We manage about $20 billion across a fund platform that includes our venture group. We're now investing out of our, I guess, OPI 8, our 8th fund, which is over $1 billion fund. In that context, we invest from a global perspective in a stage and area agnostic fashion, including but not limited to creating companies ourselves, but also investing across both the different stages of funds, but also the different cap structures of companies as well. So glad to be here and look forward to what promises to be an exciting panel.
Thank you, Peter. Sarah?
Thanks, Jay, and thanks for the Sachs group to organize this and invite me. It's great to be here with you all. Sarah Bhagat, been with Sofinnova as a general partner for some time now, was a neuroscientist for about 10 plus years before joining venture. And so started my career at Mass General doing clinical trials in psychiatry, was at Rockefeller for a few years doing more basic research in neuroendocrinology with a partnership with Seabrook. Was then did my PhD in neuroscience at Yale, focusing on molecular mechanisms of regeneration, did a brief postdoc at Stanford, focused more on basic mechanisms of synaptic plasticity. Sofinnova has been in the life sciences for a long time. We were founded in 1974. We're investing out of our 10th fund now, which is a $650 million fund, more focused on mid to late stage investments. So we're a little later stage than groups like 5AM, but we certainly love to co-invest with them on some of their earlier investments, which we've done historically. We invest exclusively in therapeutics, so really just focus on drugs and drug development. Mostly clinical stage investments, about 70 to 80% of our portfolio are in that stage. We're therapeutic agnostic. We really invest across the space, but really, have enjoyed investing in some key investments in the CNS space as well, with a focus on clinical execution. So we've had in our active funds alone, about 20 drug approvals, and have about 3.5 times clinical success rates to the industry average, which really leveraging our own kind of drug development experience internally from folks who've been in the industry for decades, and other colleagues as well. But glad to be here today.
Great, thanks, Sarah. Meredith?
Yeah, hi everyone. Thanks for having me as well. I did this panel, I think maybe a year or so ago, and really enjoyed it. So appreciate the opportunity to come back again. So I'm Meredith Fisher, a partner at Mass General Brigham Ventures. For those of you who are familiar with us, you may have formerly known us as Partners Healthcare. For those of you who are not familiar with us, we are the largest group of academic medical research hospitals affiliated with Harvard Medical School. And the fund was created back a little over 10 years ago, essentially to be able to take part in equity investments, as well as also originate companies that can be formed on the basis of discoveries made by the researchers and clinicians in our system. Give you a little bit of a sense of that magnitude. We have close to 7,000 researchers and clinicians, and on an annualized basis, bring in about $2 billion worth of grant money. So that's sort of the zero cost of capital pipeline that we fish out of in order to originate and identify deals. My own background, I'm trained as a PhD. Unlike it seems like a very heavy neuroscience set of colleagues here, I'm actually an environmental microbiologist by training. So every day is sort of a new opportunity for me to learn more about human biology. But as you can imagine, with that depth and breadth of a system, we invest, like most of the other folks here, broadly across modality, broadly across indication, and are largely agnostic to that, other than we do have a very strong strategic alignment with wanting to be part of new biology, new therapeutic interventional strategies and the like, driven by the fact that we sort of sit in an area in a community of discovery biology. And so that's an important component to the way that we think about making our investments. My own sort of role at the fund is a little bit split in that we will do more traditional sort of standard syndicated investments, which is terrific. And I think we mentioned that many of us have invested together, which is great, as well as also originate things ourselves. So we will actually identify and bring companies and opportunities forward, and even on occasion take part-time roles as executives there. And so while I spend most of my day as an investor, I do also have some operating roles as interim founding CEO, et cetera, for some of the companies that we start on. And we've been really active and really interested in the neuro space, largely driven by sort of both the discovery opportunities that we think exist, driven by some of the failures, unfortunately for us, I think we'll probably talk about during this fund or during this this panel, and very excited about the opportunities to put more and more time and more capital to work in this space.
Excellent. Thank you, Meredith. And John.
Hello, my name is John Gustafson. I'm with AbbVie Ventures. I've been with AbbVie for approximately almost 8 years now in venture capital role. AbbVie Ventures remit, we're really in the CNS. We're really looking at neurodegeneration, neuro inflammation. We only invest in preclinical companies. We won't invest in clinical stage companies. We're looking for really cutting edge novel science. Our normal investment thesis is really, we look to invest around mid-single millions, 5 to 10 million for our first investment, and we'll invest up to 20 million over the life of the company. While our initial investment is preclinical, we will follow the company all the way through the clinic to its IPO or acquisition, however it ends up. Our main geography is US and Western Europe, and I really look forward to speaking to the panel today. Thank you very much.
So with those introductions complete, we'll jump right into the questions. And we thought we'd start off with kind of a broad, big picture question, just getting your thoughts on the development landscape in CNS therapeutics. There are a lot of exciting new things going on in neuroscience, but the last 5 or 10 years have been a little bit of a rocky ride in certain areas as well. So any insights you could share with us? Sarah, would you like to get us started?
Absolutely, that'd be terrific. No, I think the past 5 or 10 years, we've certainly seen an increase of interest in this space. And you've seen that with increasing numbers of biotech companies, from the Denali's and Electors of the world to smaller biotechs, and pharma sort of re-engage their interests as well, which has sort of been a wavering on and off with CNS because it has had challenges in the history, a number of failures, but probably, a space with huge unmet medical need for things ranging from neurodegenerative disorders like Alzheimer's and neuropsychiatric disorders like depression and schizophrenia. And there's been little innovation on the front, but I think what's exciting is we're in a place where that seems to be changing. From both a technology standpoint with innovations in the gene therapy world, RNA therapies, protein degradation, oligotherapies. So that's an exciting new way to tackle some of these diseases. But what has been really great to see is that this is an area where clinical development is we're starting to get smarter in how we run these trials. How do we select the right patient populations? How do we think about the right endpoints for some of these diseases? For example, in psychiatry, where they've been incredibly subjective, hard to measure and quantify, how do you really measure a benefit to a patient? And I think we're starting to understand how to do this in a more sophisticated way, which has brought back some of the older targets like muscarinic receptors for companies like Karuna Therapeutics that really saw some exciting data in their phase 2 on both positive and negative symptoms in patients with schizophrenia. And I think it's really just sort of better understanding how to run these trials and bringing back some of these targets that we know are effective, but just not understanding which patient populations, which I'm sure we'll talk about more, but personalized medicine has really become kind of the forefront of how do we begin to tackle some of these diseases? How do we get smart about them? And then how can we extrapolate from a smaller patient population to really make an impact in larger diseases like Alzheimer's?
And maybe just to add to that, I think you hit on a key point that, you know, I look at this as kind of neurology is going kind of the way oncology was 10 to 15 years ago. We really are, as you said, the genetics are starting to come together. We're starting to build the pathways together. And I see, you know, I was looking at a company recently that's looking at patient stratification in Parkinson's and Alzheimer's disease, which is fantastic because we've never really been to there. I think we still need a little bit more on the animal models to get a more predictive models. But combining that with the genetics, I think we are getting to the point where we can develop much sounder clinical trials and have a much stronger hypothesis of what we want to test.
Great. Anyone else like to jump in?
So to build on what Sarah was saying around getting smarter around the clinical trials, I fully agree with that statement. If we look at what's been developed over the last 5 to 10 years in terms of both PET tracers, also the biomarkers that we use to measure target engagement like neurofilament light, and also the human genetics too, has really given us a very crisp understanding of what types of approaches we should be taking in these different patient populations. But I think there's also another avenue here that's very exciting when you look forward into the next 5-10 years that will change the way we do things, and that's digital health solutions, particularly when you start thinking about real-world evidence and data collection to inform not just the clinical trials, but also how products perform in the real world. And that type of data can really inform how we think about new approaches to diseases, particularly when we start combining those insights with data we can get from the commercial databases around the impact that drugs have on broader risk factors for things like dementia.
Great, thank you, Guy. Would anyone else like to add anything?
Just to kind of pile on to one of the comments Guy made, you know, part of the evolution of how we've done clinical development or how that's changed over the past 10 to 20 years has been, you know, an increasing emphasis on PD-driven drug development, where you can develop confidence relatively early in drug development that you're actually on target, on mechanism, and putatively testing the biologic hypothesis that got you interested in the first place. I think we're still in early days in being able to apply that to most CNS disorders. But I think that, you know, you're starting to see the availability of technologies and approaches that allow us to put development in this field kind of on the same footing as it's been in some of the other therapeutic areas. Additionally, as Sarah noted, and I would also underscore that, you know, this is an area where, you know, we haven't had a lot of innovation, broadly stated, in how we measure the impact of therapeutics in these diseases. Many of these, you know, validated scaling systems are very old. We know that they're incredibly insensitive. In many cases, don't directly measure patient benefit, and Guy touched on that as well. And I do think that's going to be an area for continued collaboration between academia, industry, and the regulators as we, you know, seek to try to define better, you know, endpoints for our trials that are more directly related to patient benefit.
Great. So I think we're going to dig in a little bit more detail now, because you guys are obviously spending your money. So we just wondered, kind of for the audience really, which areas do you think are becoming more exciting in the CNS? There's obviously older areas like neuropsychiatry, there's newer areas like neuro inflammation. And underneath that, there's whole swathes of cells that I'd never heard of 10 years ago, like oligodendrocytes and, you know, microglia and astrocytes and things. So I don't know, you guys have to sort of pick out some of these opportunities at quite early stages. So we thought we'd sort of dig into which areas you guys are finding exciting. So maybe I'll start by picking on Meredith. You know, you've got, you know, thousands and thousands of PIs sending you patents and publications. How do you go about sorting out what's interesting and which areas are coming forward?
Yeah, I think it's a great question, Tim, and thanks for asking me to speak to this. You know, to pick up on one of the areas that you spoke to, I think neuroinflammation certainly is a place where we've been spending a lot of time in thinking about, because I do think we have started to understand, let's say, more of the interplay between the classic CNS cells that we have sort of all known that are sort of part of the dominant paradigm, and the interplay for that with actually the inflammatory processes as well, even also immunologically as well. And I think starting to tease some of those out has allowed us to further refine and further understand the way that we might want to intervene there, as well as also trying to tease out that inflammatory component and how important it is early on in disease versus later in disease. And so I can say, even from the early sort of discovery biology side, we've seen a lot of really exciting things come out of our research basis, as well as also then on the clinical side, being able to actually figure out how to map that specifically when we're actually asking questions, either of patients and I think importantly of biomarkers. So I think that's a place where we've spent a lot of time and seen a lot of things become quite exciting. And then also, as we're thinking about new modalities, you know, having sort of genetically defined patient populations where we think that we can apply some of these new and emerging modalities, like such as gene therapy, also provides a really exciting and interesting entry point for us. And so I'd say that reflects in our most recent portfolio. And I think what you'll see moving forward is sort of a transition for our portfolio into some of these other sort of more biological driven phenomenon that we need a more robust understanding of the biology to come up with those interventional theses and strategies.
Thank you. And I know, Jamil, you incubate companies from a very early stage as well. Maybe must be tough to choose what to incubate. So maybe you could cover, you know, what your interest might be and how you go about that.
Yeah, no, absolutely. And I think very similar to Meredith, I'll second the neuroinflammation side. There's really just been this explosion of data related to human genetics to increase our broader understanding of disease pathophysiology. And so we really try to follow the human data, whether it's genetics, human primary tissue, to inform the different areas that we're going into. And that allows us to then focus on whether it's a rare disease or a subpopulation in some of these much more heterogeneous patient populations to get to a higher probability of technical success as we think about getting molecules into the clinic and actually addressing the underlying etiology of the disease and correcting that, whether that's with a small molecule, a large molecule, a genetic medicine, you know, nucleic acid, gene therapy, gene editing, cell therapy. It's, you know, there's the entire kind of tool set has increased dramatically from that perspective as well over the last couple of years.
Right. And maybe Guy, I'm going to put you on the spot. Obviously, a bit like Lilly, J&J, historically, we've been the guys doing those big late stage phase 3 trials. What do you predict is going to be coming forward that's going to excite you in the future?
I think there's a lot of potential in the heart when you start looking at the equity markets now and how that's played into the development of a lot of new companies in neuroscience over the last 3 or 4 years. You know, when I first started at J&J, one of the metrics I used to use in terms of evaluating companies was have they raised a reasonable Series A? You know, and in those days, a $10 million Series A was a pretty good raise. Now we're seeing companies bringing in $50, $60 million, and so it's actually changed the sort of landscape a little bit for us in terms of how we look at companies, because many of these companies now have the wherewithal to be able to take these products through to a phase 2 proof of concept. And so what I'm excited about in terms of the sort of areas of interest is the breadth of targets that people are pursuing, particularly in the neuroimmune space. We've learned a lot about the biology around disease-associated microglia. We've learned a lot about the role that neuroinflammation may play even in neuropsychiatric disorders like mood disorders. So I think there's a lot of potential there. We're learning a lot from other fields too, because obviously some products that work in the periphery also get into the brain and also impact the neuroimmune system. So I think it's the diversity that I'm most excited about, because I think with these multiple, very reasonable, very solid hypotheses that are being built around the human genetics, we're going to see some very significant advances in areas, particularly the neuroimmune space I'm very excited about, but also to build up some of the conversations earlier about different modalities. We're now seeing technologies coming to the fore that allow us to address targets that were previously considered undruggable, simply because we can use ways of intervening at the level of RNA and gene expression So to me, that new area of biology, when you start thinking about the breadth of different load risk factors, means that we're probably going to see a lot more clinical data coming from those types of exploratory clinical trials in very targeted patient populations. Now, while we're very much invested in the larger disease states, we are very interested in some of these mechanisms that may be developed in orphan diseases, particularly where we might be able to pivot from the learning that can be gained in orphan disease to that broader patient population. And this is an area that we would love to work with people who have assets in that space. And again, those earlier orphan type clinical trials are going to mean we're going to get clinical data back and also target engagement data back that can help drive our interests and clinical plans for other studies that may be of relevance for that target.
And I think building on that a little bit from both Guy and from Peter's comments, in neuro indications where the disease progression timelines are so long, it's that critical to have those markers of target engagement to every step of the way increase your confidence that you're testing the hypothesis and funding further programs and that you'll actually get an answer at the end of the day of this works or it doesn't work, given those timelines.
Excellent. Another area that we wanted to touch upon in our discussion today are some of the challenges in CNS drug development, from translational work and finding predictive animal models to drug delivery, especially with new modalities, including things like gene therapy, and then even more basic challenges like the placebo effect in clinical trials. All of these have been issues over the past few years. And maybe Jamil, based on your work at Sage, I hate to put you on the spot again, but if you want to comment on some of the more difficult challenges that you faced, and what do you think the approach will be going forward?
Yeah, no, as you think about neuropsychiatric diseases, it's probably one of the hardest areas to do clinical development. Long timelines to see an effect, subjective endpoints, and preclinical models that just do not represent the human disease. And our CEO at Sage, Jeff Jonas, always said, sooner or later you have to put the product into patients and ask the question. So there is always, to an extent, that leap of faith. As we've thought about it at 5 a.m., I think we try to triangulate with leveraging those human genetics, leveraging human tissue samples. Now, I think with increased tools related to iPSC, neurons, microglia, oligodendrocytes, astrocytes, you name it, we can co-culture cells to have a better understanding of what's potentially happening in vivo. I think there's still a big gap on the translational side in actual in vivo disease models. Even for rare genetically defined diseases, they still have their limitations about thinking about dose response and scaling to humans. But I think we're getting better and better. I think on the ability to measure biomarkers in humans, I think that has also progressed very dramatically. And so getting to a rapid proof of activity, as I say, in an early study, in a patient or even in a healthy volunteer of just target engagement is really critical in those early days of clinical studies.
Excellent. Thank you, Jamil. Sarah, would you like to weigh in on that?
Yeah, absolutely. I certainly echo what Jamil is saying. When I was running clinical trials at Mass General, these were mostly phase 3, phase 4 trials. Patients with bipolar disorder, the endpoints are really challenging here. And I think we're getting better at how to think about them, but even how we diagnose these patients is somewhat, in psychiatry at least, is pretty arbitrary. There's not a lot of research or evidence around some of the key criteria. And you put these patients into a box that then puts them down to a treatment paradigm, which really just doesn't have enough research and evidence around it. I think we're starting to think of creative ways to become less subjective in some of these quantifications. How do we diagnose patients? How do we predict what's the right treatment paradigm for them? How do we deal with some of these placebo effects as well? And I think there's no detail too small to really execute well. There are certainly teams, for example, you know, with Sage or the Karunas of the world or others, there's really teams who execute with great precision. I think that's going to help overcome a lot of these key challenges. The preclinical models for both neurodegenerative disorders and psychiatry are pretty awful. So you have to understand exactly what aspect you're investigating. I think the ketamine story is a really kind of key story of how there's been innovation in psychiatry. I mean, this was literally an incidental finding in 2001 at Yale with patients, you know, getting a low dose of ketamine and then all of a sudden feeling this kind of euphoric and then antidepressive effect. And for someone who started their PhD focused on molecular mechanisms and signaling transduction of ketamine and mTOR in mice, I'm confident that if that story had started in animals, there would have never been a clinical trial for ketamine. And so there's definitely a lot of these stories in neuroscience where there's sort of these incidental findings in patients. We're seeing a lot of this with some of the psychedelic therapies. And I don't know where that ends up going, but I think a lot of this is kind of empirical. And then we start to get more research-based and really understand the mechanism of action. But that has been the story. And so I think we definitely need some better models and some creative ways to think about what are going to be the biggest innovations for patients and how do we measure that.
Great, thank you, Sarah. Would anyone else like to add anything?
I think I'd pick up on one piece that Sarah sort of ended with, which is thinking about sort of something that had to originate essentially in humans to then go back and be retested in humans, and how we're actually identifying and validating opportunities. You know, from sort of our investment mindset, we expect that anything that we invest in neuro is going to have to be funded all the way through clinical proof of concept. We don't hold that same mindset with many other indications that we invest in. And so I think some of that then comes back to how do you form a preclinical data package that can convince you to place your money, right, on those opportunities. And then exactly to the point of sort of how do you design those clinical trials to select patients? Can you be biomarker driven? You have to have human data somehow pointing in some direction in order to sort of generate an investment thesis there. Because again, we just expect there's going to possibly be a lot more zeros added onto those companies before we may potentially see an exit or an opportunity for partnering. And you know, interested to hear maybe from Tim and Guy as to their views on that thesis around when they like to get engaged and what they look for.
Tim, do you want to go first?
I can start. I try not to take up too much time as the co-chair and leave most of it to you, Guy. But yeah, I mean, neuro companies, to de-risk them fully, it is a later stage game. I mean, you can have a great package, a pretty decent molecule, some biomarker changes. I mean, Lilly's very focused on, you know, disease modifying neurodegeneration, which, you know, we have highlighted a few of the issues in that space, and it's taken us, we have had some successes and some failures like many, but I think it's a complex disease with models have issues, signaling pathways in the brain and aren't always completely well understood. I think the world's getting better. This is going to move us on to our next question in a minute, but yeah, in general, to convince senior leadership to find the money to run a late stage, large indication trial is challenging. Even companies of the size of Lilly can't do, you know, too many of those. We are trying to choose smaller, faster degenerative diseases that we can use to maybe start de-risking that. But I do agree with you, Meredith, that it's not as simple as running a 4-week psoriasis trial or a, you know, maybe in an aggressive cancer where patients die within, you know, a short period of time. Sadly, it is a bit easier to pick up, you know, early signals. So I definitely agree. I don't know, Guy, if you wanted to add to that.
Yeah, it's clearly a big challenge for the neuropsychiatry field, in that the animal models are deficient. And so we do look to, as Sarah's earlier point, clinical data really to give us confidence in mechanisms that we're going after. So one of the reasons to believe, and it does become a challenge. We also use the criteria of having solid evidence for target engagement to know that we're engaging the appropriate targets and have the right biochemical endpoints. to show that we're moving the needle in the right direction. But at the end of the day, probably the big challenge we're facing is a very heterogeneous patient population, particularly when it comes to something like depression, where the etiology of it is very different from one patient to the next. And so to lump everyone together for a clinical trial is not the way to go. And equally, we're labeling a pathology or disease state with tools that are very rudimentary and go back decades, whereas we now know that there are subsets of these diseases that probably will respond in different ways. So, for example, anhedonia, cuts across many different disease states, but there's probably a very discrete circuitry that's involved in that particular symptom. So we need to think beyond the classification of diseases into thinking more about the neurocircuitry. And this is where I think we face a big challenge, because clearly a human brain is wired in a very different way to a rodent. So when you're trying to map changes in behavior from a rodent to a human being, there are limitations in what you can accomplish. So there's no magic bullet to this, but we just need to be cognizant moving into those studies that we're asking the right questions. If you go back 10 years, we were lumping everyone together into an Alzheimer's disease classification based on neurological constraints. And we now know that over 30% of the patients who were enrolled in those early amyloid-based clinical trials didn't have amyloid in the brain. So we weren't even asking the right question of the patients who we were treating. So I think this is where there's a lot of promise in the field, in that if we can use these biomarkers more sensibly and also look very carefully at patient segmentation to personalized medicine, this is definitely the way to go. And sometimes a higher power study in a smaller patient population is going to be much more informative than trying to do the sort of all-comer type clinical trials.
Great, thank you, Guy. And John, maybe if you'd like to add your perspective.
I think, overall, I completely agree with everything being said. It's in my mind, really, when I'm looking for investing, if there's anything animal models that can really help inform, we've been looking at that for a long time. Obviously, you look at oncology, they can do explants and they can do all this, and they have animal models are predictive. We've been curing mice of cancer for over 40 years now. And in the neuroscience space, it is just the animal models just are not predictive at all, as you've just been hearing. So I'm always looking out for new, unique models that can have some sort of prediction. There hasn't been any broad-based yet, but that is one of the key areas for me that I always want to look at and just see what kind of animal models are they bringing that to the table, because I do like to look at companies that have unique animal models and that can hopefully be somewhat predictive to take it into the clinic.
Thank you, John. So that brings us on to our next question. I guess we'll go right back to the other end, maybe with you, Peter. I guess in investing and forming companies, you see lots of terms banded around and kind of hype around certain concepts, and we are in the era of, I guess, precision medicine. So I just wondered what your thoughts were around, does that even apply to neuroscience? I mean, I guess you're probably used to forming a lot of oncology companies as well, and the genetics obviously is first and foremost in many of those approaches these days, but is that ever going to work in neuroscience? Is that something at Orbimed you look at closely, or are these diseases just too complicated and it's not really going to be helpful?
Well, I'll give a couple of answers to that question. One you know very well, which is Orbimed, at least in select cases, Prevail being a good example, which is now a Lilly company. It pays a lot of attention to genetic credentialing of targets and intervening in kind of a bespoke manner that's matched to the genetic lesion. Having said that, I would say that, we've spent a long time in oncology going from, defining disease by 19th century German light microscopy to the first tissue agnostic biology driven indications for drugs. That was a long time. And one of the enabling characteristics of precision medicine and oncology that I think represents a challenge in what I call the attempt to oncologize everything is that somatic mutagenesis in oncology is not the same thing as a germline genetic alteration. To pick one of many reasons why that is the case is that both the mechanistic and temporal link between the somatic lesion leading to neoplasia is relatively firm and can be quite firm, and we've been able to intervene successfully there. The same arguably cannot be said about, for example, a synuclein missense variant in a population leading to a higher likelihood of Parkinson's disease in the 5th or 6th or 7th decade of a patient's life. And in that context, it's not necessarily obvious that rectification of that genetic lesion in that 50, 60-year-old patient will necessarily lead to reversal of the pathology. That being said, I think that this is a time where we can and should test a range of those biologic hypotheses. I don't know how they're going to turn out. We've got our portfolio company, Escape, has the first mutation-selective CNS penetrant LRRK2 inhibitor. That is precision neurology. And represents a testable hypothesis in the clinic about intervention in that context. One can also take an agnostic view of that, for example, our company Aspen, which has developed some machine learning approaches to finally define purity and potency for iPSC-derived neuronal precursors. You can use cell-based therapy to try to kind of reset the clock, so to speak, with respect to neurodegenerative disorders in a manner that you could argue is no longer precision medicine. It's just, an attempt to repopulate a part of the brain with cells that have subsequently dropped out. But I think broadly stated, it's incumbent upon us to see if we can follow that evidence and marshal all of the tools that have been referenced by the panelists to test these biologic hypotheses in the clinic in a sufficiently robust manner that not only our companies but the field can trust a negative result as reading on the biologic hypothesis and enable us to move on. This is, I think, challenged in part by some of the comments that Sarah and Jamil and Meredith and Guy all made, which is, in getting back to oncology as a reference point, we are still kind of in the days back when I was originally training at NCI in the early 80s. That was a long time ago. But in any case, where there was a concept of, for example, breast cancer. Now, if you look at how we think about that, getting to Guy's comments about, depressive disorders, et cetera, we don't think about that anymore. We think about this in the context of the underlying biology that's driving the pathologic process. How close are we to being able to do that from a CNS perspective in terms of identifying the circuitry that's disordered or the areas that are disordered or the cellular processes that are disordered that likely operate, across phenomenology that are broadly applied clinically? That's going to take a while to, those mindsets are going to take a while to change. But I think, sorry, Tim, that was a long-winded answer to your question, but I think we're kind of on the cusp of being able to make some advances in our understanding, and I think the advances in our understanding are going to come from clinical research.
Thank you, Peter. That was a very thorough and thoughtful response. I appreciate that. And probably got time for one more person to weigh in, just so we get through a couple more of the questions as well. So anyone else want to cover the excitement or lack of in genetics in neuroscience?
I would maybe just say one comment I think around some of these advanced modalities, which are really exciting, is that we're often trying to see a path to that beachhead indication or rare disease population, and then the expansion to the broader population, because sporadic AD, PD, ALS, they're very heterogeneous, they're very complex diseases, and there are huge unmet needs in those areas. And so what we try to see is that permission to believe that once you get the approval or the proof of concept in that beachhead indication, as we call it, that you can then run those larger, more expensive, longer clinical studies that are hopefully de-risked at that point.
So as you look at some of these exciting new modalities, RNA-based therapeutics, ASOs, gene therapies, gene editing, protein degraders, how do you weigh the risks and potential rewards of these relatively unestablished new modalities versus traditional small and large molecule approaches, particularly when making investment decisions? And maybe if one or two of you could comment on which of these new modalities you're most excited about. Meredith, would you like to get us started?
Sure. I mean, I think, maybe the short answer is we build a portfolio. And so within a neuroscience paradigm, we want a portfolio approach that reflects different types of modalities, different types of indications, and different risk profiles. I think where we've had the most comfort and interest are probably for new modalities in the gene therapy space, and then for, let's say, tried and true modalities in the small molecule space, simply because I think we have a lot of faith that we can understand and build chemistries that can be blood-brain barrier penetrant, that can be potent and specific. And then we're relying on, did we actually get the therapeutic interventional strategy right? For something like a gene therapy, where you can envision a genetically defined population, so you have perhaps even in an in vivo model, some maybe a little bit more conviction of efficacy there, because you can potentially recreate that that error, that mutation in there, and see similarity. There, we probably want to take, let's say, less risk into something that would be completely new versus genetically defined, when we're sort of bringing something like a gene therapy forward, where there's still a lot of excitement and a lot of opportunity, but also a lot of challenges to solve in that modality, all the way from design and manufacturing to delivery, trophism, and expression levels on a per cell basis, and many others. So I'd say that that's the way we think about it at a portfolio level, and then matching that risk profile to something that's slightly less risky for us.
Great, thank you, Meredith. Sarah, would you like to add to that?
Yeah, absolutely. I mean, I think with each new modality, you have to be solving an unsolvable problem, because there's always issues around manufacturing, the regulatory path, just safety issues and concerns, right? And so we have to be able to address all of these challenges. We've done, Sofinnova's done a number of gene therapy investments. We've done about 6 to date, including Spark, which got one of the first gene therapies approved. And certainly for CNS, I think there are a number of indications that circuit specificity, for example, really matters. And so you're not going to be able to address some of those issues with a large or small molecule. So I think the best analogy, to put it is sort of when your car's out of oil, if you open up the hood and sort of dump oil all over your hood, sure, you will get some oil where you need it, but overall net effect, you will screw up your car pretty severely. And so I think we know that about CNS, right? For a number of years, we've known for indications like depression and Parkinson's, we need more serotonin, we need to modify dopamine. But if we just dump this everywhere, the net effect isn't always good for patients. And so I think there are going to be indications where you really want maybe a promoter-driven approach or cell type specificity. Something that I'd say, when I was in lab making a somatostatin specific AAV or a parvalbumin specific AAV was super easy. There's a bit more challenge when we try to translate that to the clinic. And so I do think these innovations are going to be very important to get to the problems that we're not going to get to otherwise. But It's going to take time for us to get there with a lot of these. And so they have to be serving an unmet need. It can't just be cool to be cool.
Yeah, I would say a decade ago, probably most commercial analyses would say that chronic intrathecal delivery of a nucleic acid wasn't commercially viable, right? And I think we all know how that played out. And so as we think about these applications in really rare or devastating diseases for these advanced modalities, I think where we're trying to make that jump as well to, are there some of these more common diseases that are more asymptomatic, more slowly progressing? Are people going to be willing to take those types of therapies with the risks associated with them? And I think back onto the medtech side of things like Parkinson's, where deep brain stimulation actually works pretty well in the clinical studies, but very, very few patients actually get neurostimulators implanted in their brains out of just kind of fear of the procedure and the risks associated with it. And so gene therapies and ASOs and cell therapies bear those same concerns from the patient population.
I would like to pick up on one point that Sarah made, which I think is probably the next great frontier in terms of thinking about how to intervene intelligently in CNS disorders. Certainly we go through this all the time from a neurosurgical perspective, but it is no less true generally in neurology that kind of like real estate, it's about location, location, location. And I loved Sarah's analogy about pouring oil on the car. When you think about it, we're kind of bathing the CNS in a chemical. At some level in that context, you can see the small effect sizes that we've achieved in things like depression and say, wow, that's actually pretty impressive given how flawed the approach is. But if I was going to pick one of the areas that would be most exciting to see developments in, it would be the abilities to localize therapeutic interventions to disabled circuitry.
Great, thank you, Peter. And anything that you'd like to add, John?
From AbbVie's perspective, when we're really looking to invest in areas that we consider kind of complementary to our R&D, so we're blessed we have a lot of small molecule and other approaches going on, but I would say in general, everything that you mentioned, the RNA, the protein degradation, gene editing, gene therapy, are all areas we're looking at from an investment point of view. We also kind of look at this from a portfolio approach and how does this complement with our internal R&D. And so I'd say, what we're really looking at is a novel science and what is the hook? What is this bringing that no one else is bringing to the table? What hypothesis is this company going to be able to interrogate, and what can we potentially learn from that? That's really how we're kind of looking at it. So we look at it kind of in a broad overall portfolio. We would never invest in something where we have internal research, just a small, maybe a small molecule approach going after tauopathies where we know something. Just as an example. But what we will look at is, novel areas such as maybe a gene therapy or protein degradation, something that we're not going to take a look at internally. And that's really how we want to look at our portfolio and how we do our investments. I think it has to have a strong hypothesis. And then with all ventures, it has to, why this biotech company should we invest in versus someone else. What's their special hook that they can do that no one else can do? And we're not afraid of looking at really risky areas as long as there's a solid hypothesis behind it and a good solid team that can say, we can do this. We have something special that no one else can do. That's really how we like to look at our investments. And all the areas that you kind of mentioned are all areas that we're very interested in taking a close look at and investing into.
I guess simplistically with investors, many people would probably think it's all about therapeutics and the molecule, but I guess in neuroscience, there's a whole load of other areas that are becoming more and more talked about. And I just wanted to probe whether they make good investable companies. So we've got, I think, Peter, you mentioned AI and machine learning approaches. Many people have talked about drug delivery, patient diagnostics, even new modalities as just molecule companies even. Are those sorts of, any of those areas, the sorts of things that you would actually create companies around and then apply them to neuroscience? Or is it really all about just the biology and the molecules interrogating it? So I guess that probably would open up to most of you on the panel, but Jamil, why don't you kick us off?
Yeah, no, happy to step on a soapbox here and talk about precision digital therapeutics or prescription, sorry, digital therapeutics, where we funded a company called Pear Therapeutics back in 2012, 2013 to develop software to treat human diseases and apply the rigors of randomized controlled trials to that development to justify physicians prescribing, patients using, payers reimbursing for products. And we see that as a new modality kind of right next to gene therapy, cell therapy, small and large molecules going forward, with now Pear having multiple approved products, having their own commercial infrastructure and launching those. And so I think it's an exciting area that many, many more companies have now gone into and raised capital, advanced programs in the clinic. So it's an area to watch for sure.
Great. I'll open it up to everyone. Just I guess we're sort of coming towards the end, but quick fire. Are these sorts of technologies attractive to you guys to invest in, or is it sticking to therapeutics? Maybe Meredith?
I would say it's attractive to us, but one of our biggest concerns, particularly if we're thinking about AI or large volumes of data to analyze, is how good are those data? And I think we touched on some of the challenges in generating data that we feel are robust and solid enough that we would have a strong conviction. So I'd say, yes, we're interested, but our bar is quite high, and we probably think that's a few years off before we'd have the conviction.
Sarah?
Yeah, I mean, Sofinnova specifically is focused on therapeutics. So we're really just focused on drugs and drug development. You know, I think we specifically do like to see some proof of clinical data and evidence. That being said, I think we need some of these tools to come to market for us to be able to successfully develop some of these therapeutics. So certainly when we've seen companies that have a really kind of innovative approach to redefining an endpoint, certainly we want those to be a little further along, but that's going to be absolutely necessary to get these drugs to the finish line. And so I think it's absolutely important that there's more innovation in this space, even though it's not a focus of ours.
Right. And Peter, can a fund the size of Orbimed cover all of those bases?
We view those through the lens of their applicability to therapeutic interventions. And I do think Jamil raised a great point about the emergent field, perhaps unsurprising, that training and directed training, et cetera, can and does have an impact. We've known that for a long time. And systemating that and putting that in a way that is a uniform intervention is an interesting advance for the field. But broadly stated, there and elsewhere, we look for the therapeutic application of these technologies rather than the technology per se.
And last but not least, Guy, I guess, are you investing internally in all of these different areas to be successful?
We do, and like Peter said, from the Janssen perspective, we tend to look at the lens through sort of the products that we're bringing to the market, therapeutic. But having said that, if you were to gaze into a crystal ball over the next 10, 20 years, these technologies are going to be wedded to ensuring healthcare solutions for patients. Because part of the patient journey is not just about receiving treatment, it's about the follow-up care they get, it's the remote care they get. It's also the accuracy of the diagnoses too. And so, with the advent of 5G, I think this is a really wide open space. We've seen a huge amount of investment go into digital health solutions just in the field of depression over the last 3 or 4 years. So this will expand, and we're going to be looking at solutions that can help us, obviously from the precision medicine standpoint, but also thinking about the wraparound care and solutions that patients get when they are using our products.
Anything that you'd like to add, John?
For AbbVie, our sentiment towards neuroscience, this is an area we heavily want to invest in. When we look overall at our portfolio, oncology is a key area, but then right after that, neuroscience, and that is not going to change. We make about 6 investments per year. Every year we're doing more and more in the neuroscience space. We look at this maybe not something that's immediate within the next couple of years. It's going to have a huge breakthrough, but I do look at this, come from my personal point of view, 5 to 10 years from now, our investments today, I think, are really going to make a large impact. And so from an AbbVie point of view, neuroscience is an area we're definitely going to continue to invest in. And, along some of the modalities that we talked to and others that probably we haven't even thought about yet that'll be coming. But from our sentiment towards neuroscience is definitely an area that we will be active in for the foreseeable future.
Great, thank you. I'm not sure if we have time for maybe one more question, maybe a lightning round. Just trying to test the investors' sentiment towards neuroscience investment these days. I guess, we've talked about some of the sort of binary risks associated with investing in neuroscience. I know, Meredith, you talked about the portfolio approach to diversifying some of that idiosyncratic risk. Could you maybe just talk about your the key drivers of your investment thesis in neuroscience? I don't know, maybe Meredith, if you want to get us started.
I think for us, we have to have conviction that the interventional thesis is as robust as sound as possible. I think that comes down to, is there human data supporting that? Is there a biomarker or a path towards a biomarker or some sort of early readout that would give us clinical conviction? Do we think we can either move to something that is a small indication that could be broadly expanded, or to use that biomarker or some other agent to have a patient selection criteria? And then to balance sort of the risk of modality with all of those other risks to sort of provide us enough support to step forward and to deliver on that. That said, I'm quite, while our bar has to be high because it's a lot to do and a lot to be solved for, I think it's an area that we're also probably some of the most excited about, unfortunately, because of the significant unmet need that remains for patients, but I think importantly, because there's still a lot more that could be done here.
Great, thank you, Meredith. Sarah, would you like to add anything?
Absolutely. I mean, I think, what Meredith is saying is spot on in that there's, for us, kind of key investment thesis would be that there's a huge unmet medical need. And I think if you look at most CNS indications, you hit that bar is fairly low. I mean, there's just such a huge unmet need. We don't believe for CNS things have to be disease modifying, and these patients are struggling. They have huge issues with quality of life, and symptomatic relief is significant for them. And that's enough for us to get conviction. I think the hardest is that, from phase 2 through approval, there's about a 14% clinical success rate for CNS. It's really relatively low. Ours is about kind of 3.5 higher than industry average. And so there has to be, key endpoints or clinical data in existence to really help us believe that we will help these programs move forward to approval. And so there has to be a real time frame that we're able to really help move these therapies into the hands of patients and clinicians, because I think it's a tough space, and we're really trying to make innovations for patients. We, as Sofinnova say, we fund drugs and not dreams. And so we do try to look for good data points that we have high conviction for these therapies coming to fruition.
Thank you, Sarah. Jamil?
Yeah, I would just add, I obviously completely agree with Meredith and Sarah's perspectives, and would just add, we think about building strong syndicates to carry through to those key inflection points. And those inflection points are often later in clinical development. But if we, in our investment theses, we're mapping out a path to understanding those biomarkers, I think there have been good precedent transactions that larger pharmas, if you have good genetic validation of your target, if you've got an early biomarker you can measure in a phase 1 study, that they'll engage in discussions at that time. And there's some great transactions that have been announced on early phase 1 data in the $1 billion plus biobuck range. And so there is an appreciation for the needs for these patients and desire to be in these fields or indication spaces. So I think there's a lot of potential there.
Excellent. Thank you, Jamil. Peter?
I don't have much to add in terms of how to think about intelligent investing in the space beyond the cogent comments from my colleagues. Obviously, Orbimed remains very excited about the space. We've got a significant portfolio of companies here, and I think that reflects a belief that that if you do this well, there's a significant return available both to patients and to investors for innovating in a space that desperately needs innovation.
Thank you, Peter. And Guy?
I agree with many of the comments that have been raised. Probably the one thing that hasn't been touched on is the people and the team, because drug discovery in neuroscience, as everyone knows, is challenging and it has pitfalls. And one of the things we look for is a team that's very capable of addressing those issues and challenges when they do arise.
Excellent. Well, I think we're just about out of time. So thank you all so much for sharing your valuable insights with us here today. Really appreciate the discussion, and I want to thank Sachs Neuroscience Forum for hosting this event. And Tim, any closing comments?
I just hope the audience take away a very positive message that I think unanimously everyone here is excited about the space, making further investments, trying to push on biomedical research to patients. And I guess from my perspective, please, if you have interesting projects that you think might be applicable to Lilly, please get in touch. And I'm sure most of the panelists would echo those sentiments. So yeah, thanks everyone, and it's been a pleasure.
Thank you.
Thank you.
Thanks.
Everyone.
Take care.
Thanks, everybody.
Bye now.